So, good evening, guys. Am I audible to all?
Yes, you are audible, sir.
Okay. So, today we'll be starting with the discussion of the course Experimental Biotechnology. So, starting with my introduction, I am Rishabh and I am a PhD scholar in IIT Kanpur. So, I'll be taking this lecture series as part of my TA requirements. And so basically we'll keep this session as a discussion forum where we'll be discussing about different doubts that you guys have and how to solve different questions. So I have prepared the meeting, the presentation in a way that we can discuss about the, just a second, so that we can discuss about different problems and how they are asked in the paper. Okay. And I want you guys, if you guys have any query, any problem, you can stop in between and ask questions. Okay. So I would prefer to this platform to be a more discussion based rather than a lecture based, so that you guys will understand a bit more. And if some people have some language issue, they can also let me know. Okay. So let's start. And so let's start with the first question. So, and I'll be following the pattern that has been followed in the lectures. So, this is the first question. Match the following organisms with the associated biosafety levels in which they are worked. Okay. So, you have to match different organisms with the biosafety levels in which they are worked. So, please read the question and guess the answers. We'll discuss after that. So, any answers? So, any answers? Okay.
Ebola. Hello.
Yes.
Ebola virus is BSL 4, but here it's written BSL 2.
Yeah, you have to select the right option from this from the given options.
Oh,
Okay, so you have to
okay.
select the right option from these options. हेलो. हाँ बोलो. अरे यार, मैं चाबी तो मेरे पास ही है. लो. Hello. Uh-huh.
I think A3 means first option is correct.
Okay, so let's discuss. So we have first microorganism, that is uh, Mycobacterium tuberculosis strain H37RB. Okay, so this is a basically a causative organism to tuberculosis. Okay, and then we have E. coli, that is not dangerous at like less dangerous comparatively to the other organisms, and then we have Ebola and then the adenovirus. Okay, so the Mycobacterium tuberculosis basically causes tuberculosis. So it needs a safer environment to work with. Okay, and so it will require BSL-3. Then we have E. coli, because E. coli is used in our daily cloning activities and all those stuff. So we can culture it and use it in BSL-1. Okay, then we have Ebola virus, that's basically airborne. So if we are working with it, we need sophisticated equipments, and to basically just a second. So we need sophisticated instruments to tackle this particular organism. So Ebola virus needs BSL-4. And then we have adenovirus. So adenovirus is basically like less pathogenic, and it's used in various laboratories for recombinant DNA technology. So it can be worked with BSL-2 environment. Okay, so now we'll see the options. So A is 3. That means Mycobacterium tuberculosis can be worked in BSL-3. So yes, it's correct. Then we have C is 2. Then that means Ebola virus in the BSL-2. This is wrong. Then D is with BSL-2. Yeah, this is correct. And then we have B, that is E. coli with 4. That is BSL-4. So this is also wrong. So this is a multiple selective question, and the correct answer is 1 and 3. Okay, and so basically what happens with the BSL levels that we study. So BSL-1, that is biosafety level 1, is associated with the organisms that pose low risk to the personnel as well as to the environment. So they are non-pathogenic and do not cause any harm to the person that is working with the organism as well as to the environment. Then we have BSL-2. BSL-2 are associated with the organisms that leads to moderate risk and to the personnel or the environment. So they can cause but milder diseases that can be treated with the uh, normal medicines that has been prescribed. Then we are moving on to the BSL-3, which means that it causes serious disease to humans or to environment, basically to plants or animals, and they are not spread through casual contact. Okay, so that is why tuberculosis is being worked in BSL-3. Then we move on to the airborne pathogens. Okay, so which causes the disease based upon their presence in the environment. So that can harm both the humans that has been working with it and the environment as well. So that's why Ebola and all those stuffs are basically worked under BSL-4. Okay, so did everybody understood the question as well as the explanation associated with it?
Yes, sir.
Okay, now we are moving on to the next question. So what is basically material data sheet? So the options are a catalog of standard operating procedure for your lab, a catalog of standard operating procedure for biosafety cabinets, a catalog of information about the potential hazard or of the material provided by the manufacturer, or a catalog of instructions to be followed if any spillage, accident, or injury happens. So can anybody guess? Okay, so many people have said option 3 or option 4. Okay, so let's check the answer. It's option 3. Okay, so what happens is basically the material data sheet that we are talking about gives the information about the material that we are using. Okay, so this is uh, this will give the information whether it's poisonous or it's flammable, it's corrosive in nature, or radioactive, and the things like that. So it gives basically the information about how the material is like and what's the qualities of the material. Okay, so it's the catalog of information about the potential hazard of the material provided by the manufacturer. Okay, and although it will also provide all the instructions that are being present. Okay, so that will also come under the information about the material itself. Okay, so the third option is the correct answer, and here are the different uh, here are the different features that have been incorporated into the material data sheet. So for example, there is a explosive or it's a corrosive material that can harm the b- person working with it, so it must be handled with care. Then it is a flammable liquid or not, or it is toxic or not, it's fatal. That means it might cause death when ever uh, inhaled or being spilled over. Okay, or toxic to the environment. Then it will be irritant. For example, the exclamation mark is for an irritant, so that means it's whenever it is applied on a hand or something, then it leads to irritation. Okay, then it is a health hazard. Okay, or it's a carcinogen, mutagen that leads to cancer. It's flammable, or it's whenever it comes into contact with other liquids, it might cause any flame. Or the other things that are mentioned are it must be stored in dark and cool condition, as well as the temperature at which it must be stored. So all these kind of stuff, as well as the some of the manufacturers also label the dilutions, how to dilute the particular material or particular compound with using the solvent. Okay, so these are all mentioned in a material data sheet. Yeah, you will be getting the presentation after the session. Okay, so this is how the material data sheet works well. So any doubts till now? If not, then we'll move on to the next question. So the next question is, while working with the radioactive isotopes, radioactive shields made of which material out of these options are used for protection? Can anybody guess?
Lead.
Okay, so okay, so people have opted for both lead as well as platinum. So the correct answer is option A, that is lead. Okay, because few of the features are it's cheap. That's a major feature. Platinum is very costly. Okay, so it cannot be used to make radioactive shields. Then it is non-reactor. So that's why it is being used to make the radioactive shields. Okay. So let's move on to the next question. So this is regarding the laboratory notebook that we maintain in our research labs. Okay. So the laboratory notebook is important for the researchers because the options are: it helps in recording the conditions in which the experiment is performed, it helps in publishing a paper or a patent, or it helps in to discuss the various optimization steps in an experiment, or all of the above. So can anybody guess? Okay, so it's option two according to one member. Then, okay, okay. Someone said four also. Okay, just 2 minutes. There are people. Record. So the correct answer is option four. Okay. So how it helps in maintaining the how laboratory notebook helps in maintaining all of these is, for example, I'll talk my work, okay, because I am also a research scholar. So, for example, if we are planning a cloning experiment, okay, so now first we have to check whether the reagents or the materials that we require are there or not. Okay, so the first thing would be the materials or why we are doing it. Then, second thing would be, sorry, first would be why, second would be the materials that we require. Okay, so why or the objective of the experiment. Okay, so for example, if we are doing a cloning experiment, so what is the objective of that experiment? Why do we need that cloning? Okay, so for example, if you are working on a gene X, so if we have to study the protein content or the about the gene X, how it works, so then we have to clone it in another plasmid. Okay, so this is the objective of this experiment. That why are we doing that particular experiment? So this is the why. First thing. Second thing is the reagents or the materials. So what are the things that are needed for the cloning? So we'll figure it out. What are the things that we need and what at what concentration we need, and like how many reactions do we have to set up. Then the third thing will be how we'll analyze the data and how we'll perform the experiment. So first is the experimentation. Okay, so in what amount these materials will be used so that we can get an optimum uh, we can get an optimum result in minimum amount of time, or the amount of time that is needed for the incubation or any other thing. Okay, and this is the procedure or the experimentation. Then we have the fourth option, that is the data or the conclusion that we get from that experiment. So what are we getting from this particular experiment? So for example, if we have done the cloning and then we ran a gel, and we are like, this is a gel, and we are like seeing two bands. So what does these bands signify? Okay, so this is all we get from the data or the conclusion. Then at the last is the discussion, which leads to the thing that our experiment has been success or a fail. Okay, And why these are important is because if you are doing a cloning with or any other experiment, you have optimized all the conditions that are required. For example, if you are running a polymerase chain reaction, at that particular temperature, annealing temperature, you have used a particular primer. So if another person of your subsequent batches used with the same gene, then he or she can directly use that temperature to start the PCR rather than just going with the optimization again. Okay, then all these data that you have accounted for in your laboratory notebook will help you in writing a research paper after the all the experiments have been done and linking the final result of those particular experiments. Then we have the optimization steps. Okay, so the major part of our experiments is optimization. Okay, so whether we are doing any of the smallest experiment, we optimize things first, then we move on to the actual experimental things. So this point of time when we have optimized the steps and we have to repeat those experiments, then we need the optimum conditions that have been used for that particular experiment. So this leads to replicate replication of the experiments also. Okay, so this is the major information about the laboratory notebook. So any doubts, anybody? So if not, then we'll move on to the next question. So this is related to general experimentation in our lab. So while pipetting a high viscosity liquid, which of the following techniques should be followed? So the options are that take the liquid rapidly from the pipette, cut the microtip from top, use the mouth operated pipette, or use a measuring cylinder. So can anybody guess? Okay, so someone said two. Then other options. Okay, someone said one also. Okay, okay. So what happens? We have two kinds of pipettes in our lab. Okay, so one is a glass pipette, and the other one is a micropipette that is basically plastic based. Okay, so in case of a glass pipette, we have an aspirator, and that cannot take up minimum like in microliters volume. Okay, so this is the disadvantage of the glass pipette. Majorly, when the volume is large, then we can use the glass pipette accurately. Okay, when we have these biological experience experiments where we need the volume in microliter range, so in that case we need the micropipette. Okay, that has a range from various pipettes. So we have 1,000 microliter pipette that can pick up about from 200 to 1,000 microliter. Then we have a 100 to 200 range micropipette, and then we have 10 to 20 or 2 to 20, then we have 0.2 to 2. So these are the various range of the pipes. Okay, and generally when we are doing such experiments, for example we are making a buffer, so in buffer sometimes we require glycerol. or suppose we require tritonins. So these compounds are basically viscous compounds. Okay, so we cannot directly take these materials from the reagent bottles that we have. So we require, if we require suppose 10 microliter of these compounds, so we cannot use a glass pipette. So what we'll do is, we suppose this is a microtip that we use. Generally what we do is, we just cut the front part of the microtip so that the hole from which it sucks the liquid becomes larger. Okay, now this, from this hole the viscous liquid can be aspirated easily, rather than using a measuring cylinder. For measuring cylinder also the same case, we cannot use it for picking up 20 microliter of the sample. Okay, and same with the mouth operated pipette, it's generally a glass pipette, and it's not even advisable to use these pipettes while working with biological samples. Okay, there are many contaminations or even that can harm the human body also. And thirdly, take the liquid rapidly from the pipette. So whenever we are using a micropipette like this in the tip, then we cannot aspirate the liquid very fast when it's a liquid, a viscous liquid. So the first option is also correct, is also wrong. So the correct option is option 2. So everybody understood the question and the explanation that I gave? Okay, just 2 minutes. I'm putting my laptop in charging. So let's move on to the next question. So this is related to the pH that we measure. Okay. So you prepared a buffer at room temperature whose pH was 7, but the storage of the buffer was kept. But for storage, the buffer was kept at 4 degrees Celsius. Okay. Generally, we store the buffers in a lower temperature. So this is 4 degrees Celsius. Before using it for an experiment, you measured the pH again, but it was cold when you measured the pH. So what would be the pH of the buffer now? If it might be lower or higher than 7, or it might be 7 only, as temperature has no effect, or more information is needed for the question. Okay, uh, any more answers? So someone said option number 3. Okay, just a second. Someone so basically, what is the equation when we are measuring the pH from a particular pH meter? Okay, so the voltage that we get is basically, uh, So this is the equation that we have for a pH meter. Okay, and this is basically how we calculate the voltage through a pH meter when the pH of a particular solution is mentioned. So this is the voltage constant of the pH meter, and this is the voltage that we get at particular like when we are measuring the pH of a particular solution. So from this, if we opt out this, because these are the voltage parameters, now we consider this equation alone. So pH is inversely proportional to temperature. Okay, so for example, if you have, generally in biological experiments that we use, RT, that is room temperature, is the optimum. Okay, that is considered as around 25 degrees Celsius. So at room temperature, we measure the pH every time. If it's less than 25 or greater than 25, then that is considered as to be like the pH will not be accurate as it is considered. Okay, so here uh, when pH was 7, it was RT, but it was kept at 4 degrees now. So here the temperature has been decreased. So if temperature is decreased, then the pH will be increased. Okay, because it's an inverse relation. So whenever if the solution would be cold, then the pH would be higher at that point of time. If now, if the case would be the the buffer that you prepared, you incubate it at RT for some time and then measure the pH, then it would be nearly 7. Okay, so the correct answer is option 2. And yeah, this is the equation that follows the uh, basically that follows the reading of the pH meter. So any doubts, anyone? So just remember that pH is inversely proportional to temperature. This is the main focus of this question. Okay, so if you don't have any doubt, then we'll move on to the next question. So how does a pH meter work? So it works on the principle of ammeter, chromatography, potentiometer, or ion exchange. Can anybody guess? Okay. Any more answers? Okay. Okay. So actually, a pH meter works on the principle of potentiometer. Okay, not the ion exchange. Ion exchange is one of the component of the pH meter. How it works, we'll see. So, in case of a potentiometer, what happens? The basic principle of a potentiometer is that the battery is connected to a resistor, and now the same battery we are using to check the potential difference across this resistor. Okay. So now, if we check on any of the point from this end to this end to on this resistor, then that would complete the circuit and would tell the resistance or the potential at that particular point. Okay, so now the same thing applies to the pH meter also. So what happens? This is the first electrode, the reference electrode that we talk about. And the second electrode is fitted here. So whenever we touch this particular electrode to a solution, At that point of time, the ion exchange happens such that the hydrogen ions in a particular solution is measured, and then this sends a potential this leads to a potential difference between the reference electrode as well as the electrode that we are used to measuring the pH. So this difference in potential difference in these particular electrodes leads to a value that is being displayed on the pH meter. Okay, so this is how a pH meter is basically used to measure the pH of a system. Okay, so anybody has any doubt in this particular question? If you have any doubt, you can ask. Okay, so what happens is, yeah, you are right, because here the ions are involved. Okay, but what happens, the difference in the ions that we see in this particular electrode and this particular electrode leads to a potential difference. Okay, that is being transformed in the like the this potential difference is being transformed in a particular value that we see. Okay, so this difference in ion concentration between both the electrodes leads to this potential difference, and this is how it is detected in a like the meter that we see. Because the instrument that we have do not detect the number of ions that directly. Okay, so it cannot lead to the number of ions or how much ion is different from the reference electrode that we use, that is silver electrode. Okay, so it leads to a difference in the potential difference, and which leads to a value that gives the exact pH of the solution that we are using. So any more doubts with the question? If not, then we'll move on to the next question. Okay. So weighing balance works on the principle of, the options are principle of lever, law of conservation of mass, Archimedes principle, or law of conservation of momentum. Can anybody guess? Okay. Someone said option number two. Then okay, one. Any more answers? Okay. So yes, it works on the principle of a lever. Okay. How? This is the explanation. So what happens? This is a particular primitive weighing balance that we see. Okay, so whenever we keep a weight here, and here is the reference weight that we are measuring. So both of these balance each other. That's how a lever also works. So for example, this is a lever, and here you attack something, it will go down. and if you attach another thing, then it will be balanced up. Okay, so this is how the things work in a weighing balance. The same thing applies here also. Okay, so what happens if we keep on like this is a digital weighing balance. So here we keep a weight on upon a stage, and that stage basically balances it with another reference spring that we have. So there is a spring that controls this particular weight, and this is how this uh, whenever we keep a weight on this particular stage, there is a spring associated with it. So upon adding the weight, the spring basically stretch or uh, like become small. So on that basis, the weight is being like shown in the digital meter. So this is how the digital weighing balance works. So the weighing balance, the weight that we see basically is reduced on the basis of the principle of lever. Okay, so anybody has any doubt in this question? If not, then we'll move on to the next question. So the purified water from different methods are used in an experiment to measure the absorbance of a solution. Which water will give the highest background UV absorbance in the solution? So, water purified by membrane filtration or ion exchange or purified by reverse osmosis or distillation. Which of these will be applicable in this question? Can anybody guess? Okay. Any other answer? Okay. Okay. So what happens when we are purifying any water? Okay, so we don't want any absorbance in the solution because the pure water will not give any absorbance. Okay, the absorbance is there when there is impurity in the water. So it will give some kind of absorbance, but that will be minimal. Okay, but the highest of the background UV absorbance will be seen in an ion exchange method. Okay, because in case of membrane filtration, okay, what happens, there is a membrane with which has pores. So now when water will pass through it, the larger particles that we see, and these pores are of different sizes. So whenever we are using any of the pores, the all the impurities that we have stays behind and only pure water comes out. In case of reverse osmosis, like we see in our RO systems in our houses, so they use basically charcoal filters to filter out all the impurities and the ions that we see. Then the distillation method is applicable to where we have certain distillers to purify the water. Okay, but in case of ion exchange, what happens, we have certain background column, that is basically organic compounds. So sometimes what happens when water is passed through this organic compounds, some of these compounds also add in the water. So whenever we do the UV visible absorbance of this particular water, then absorbance will be from these organic compounds also. So this is why it will give high absorbance in case of when we measure the absorbance using UV visible method. Okay, because of these organic compounds that have been washed away while passing the water through these background column. Okay, so the correct answer is option number 2. So, any doubts, anybody in this particular question? If not, then we'll move on to the next question. So, the force responsible for sedimentation of suspended particles in a sample during centrifugation is, options are centripetal force, buoyancy, centrifugal force, or frictional force. Can anybody guess? Okay, someone said option number 3. Then, any more guesses? Okay. So yes, the correct answer is option number 3, and this is how a centrifugation works. Okay, so when we put our samples for centrifugation, it's basically in this direction. Okay, this is how the tubes have been hanging in a centrifuge. Okay, so now it runs at a particular speed, so it moves in a circular direction. So now what happens? For example, here we have our sample. So now the from one end the centripetal force will be directed towards the center, and from the other end it's directed towards the outside. This is the centrifugal force. This basically is because of the weight of the particular compound. Okay, so this is the reason how different particles are basically separated using a centrifuge. So because of their weight, they apply a particular centrifugal force, and this is how they are separated or pelleted out using the centrifugation. Okay, so any doubts in this particular question? If not, then we'll move on to the next question. So now we are moving on to the solutions wala part, and this is the question: how many grams of glucose will be required to make a 100 ml of zero point 0.1 molar glucose solution? And molecular weight of glucose is given as 180 grams. Please solve it and let me know the answer. Okay. Any more answers? Someone said 1.8 gram. Okay. Any more answers? So the correct answer is option 4. And how we'll solve it? So first of all, we are preparing a molar solution. So what is the formula for molarity? It is the number of moles of solute divided by the total volume of solution in liters. Okay, so here the solute is glucose, and we have to prepare zero point 0.1 molar glucose solution, where we have to find out the weight. So number of moles is basically the given weight divided by the molecular weight of the solute. Okay, so the molecular weight is 180 grams, and the volume of the solution is in 100 ml. So we have to prepare 100 ml, but it's in ml, so we'll divide it by 1000. That is the conversion factor for the ml to liter conversion. So after solving, it will be zero point 0.1 into 80, that is 1.8 grams. Okay, the simplest way. So this is the basically based on the formula that we have. Now some people also prefer to use the unitary method. So what happens, if we are preparing 1 molar glucose solution, then the amount is required is 180 grams. Okay, so if we are preparing zero point 0.1 molar solution, then it would be 18 grams, but we have to prepare it for 100 ml, not 1 liter. Okay, so 18, this would give us to 1.8. Okay, so this is the simplest explanation to this particular problem. Okay, so anybody has any doubt in this particular question can ask. So these are the general questions that are being involved, or you will be focusing in like whenever you are doing any experiment. So you have to prepare different solutions based on the molarity or the amount that we need. So these are the day-to-day experiments that are being followed in every biology lab or chemistry lab. So if no more doubts, then we'll move on to the next question. So dash is the amount of NaOH required to make 350 ml of 4 molar NaOH solution, and here is the molecular weight. So can anybody guess the answer? Okay, someone said 1. Any other guesses? Okay, one more answer, that is 1. Any more guesses? So the correct answer is option 1. Okay, so similar kind of question that we have solved just now. So 1 molar NaOH would require 40 gram of NaOH, okay, in 1 liter solution. Then we have 4 molar NaOH. Sorry. Then we have 4 molar NaOH. So it requires 160 gram of NaOH in 1 liter solution. Then we have 350 ml of this. Okay, so this would be nearly 56 grams. So is the explanation fine? Anybody has any doubt in this? Okay, just a second. This is the previous question. Is it fine? Shall I move into the next question? Okay. So any doubts in this particular question? If not, then we'll move on to the next question. So this is also a solution related question. So a buffer contains 10% glucose, 20 millimolar tris, and 50 millimolar HCl. For making 1 liter of buffer from the following stock solutions. So this is the actual constituency of the buffer it contains. Okay. Now you have to make the buffer using the stock solutions that we have. We have 50% glucose, 1 molar tris, and 1 normal HCl. So what is the correct amount of the volume that is needed with this particular stock solutions to make a buffer that contains the final concentration as this? So please solve and let me know the answers. Do you guys need any hint? Okay, let's give you a hint. So you would have heard for a formula that is C1V1 equal to C2V2. Okay, so this is the concentration and volume for one particular liquid, and this is the concentration and volume for the other liquid that is derived from this particular liquid. Okay, so keep the stock concentrations on the left side and the final concentrations that we need on the right side, and then calculate using this formula. So, any guesses or any more explanation needed? Okay, so we have got one answer, that is 4. Any more answers or guesses? Okay, so the answer is option 4 only. So let's go on to the explanation. So as I've told you, you will be using this formula C1V1 equal to C2V2. So C1 is the concentration and V is the volume. Okay, so consider this. On left side you keep the stock solutions. Okay, and on the right side you keep the final concentration that you need. For example, first we'll consider the case for glucose. Okay, we have a stock solution of 50% and we require the final concentration in 1000 ml buffer. Okay, this is 1 liter. So we need the 10% glucose concentration, that is the final concentration, and in 1000 ml of buffer, that is 1 liter. Okay, so how much amount we take out from this 50% volume, and we'll put it in the solvent that we have, maybe water or any other thing. Okay, so this is V1. So V1 would be equal to 10 into 1000 divided by 50. So this will be 200. So 200 ml of 50% glucose will be required to put it in this particular solution and make the final concentration of glucose in buffer be 10%. Okay, so this was the case with glucose. Secondly, we have tris. So we have the 1 molar tris, which volume 20 millimolar we have to make, and in 1000 ml. Okay, so here milli means that is a prefix for 10 to the power minus 3. Now the V1 would be, so this will cancel out, and here V will be 20 ml. Okay, so 20 ml of tris would be required to like from 1 molar tris, so that we can make final concentration as 20 millimolar in the 1 liter buffer. Okay, now we'll move on to the third component, that is HCl. Okay, So here we have the one normal HCl as our stock solution. So in case of HCl, the N means normality. Okay. So normality is basically dependent upon the gram equivalents. So that is a concept used for various acids and bases. But for HCl, one normal is equivalent to 1 molar. Okay. So here we'll be using 1 molar as our stock solution, and we require 50 millimolar in the final buffer. Okay. So this will also cancel out and the volume would be 50 ml. So now what we'll do is we'll add up 50 ml, then 200 ml of glucose, and 20 ml of crisp. So this will be 270 ml, and the rest would be water. This would make 1 liter of buffer out of these three components and water. Okay, so this is how we take up different solutions. Okay, just a minute. Yeah, that's why I have used a C1. If I am using molarity, then it would be M1. Okay, so C is for concentration. So if you are using molarity as your stock solution concentration, you will keep M. If you are using gram per ml, then you will using gram per ml. Or like in the case in here, we have used percentage also. So you can keep any concentration terms in case of C. Any more doubts? These type of questions like I'm discussing, these are frequent calculations that we do in our laboratory, in our practicals. Okay, so you must be well versed with the formulas as well as the how we can apply the concept in our day-to-day experiments. Okay, so if no doubts till now, then we'll move on to the next question. So this is a question based upon the dilutions. Okay, so while measuring the potassium concentration in a drug formulation, the technologist took 10 microliter of the sample and mixed it with 90 microliter of test buffer. From the resulting solution, he took 5 microliter and measured its total potassium content as 25 nanogram. What would be the concentration of potassium in the drug formulation? So what happened here is a researcher took 10 microliter of the formulation and dissolved it in 90 microliter of test buffer. That will be a solvent. Okay, making the final volume as 100 microliter. Then from this 100 microliter, he took 5 microliter and measured the potassium concentration. That came out to be 25 nanogram. So now we have to like check what is the concentration of the potassium in the drug formulation. So can anybody guess? This type of question will be helpful in our laboratory while measuring different DNA, protein, or RNA concentrations. So, any guesses or any hints required, please let me know. Okay, so we have got one answer, that is 25 nanogram per microliter. Anybody else? So the correct answer is option number one. Okay, so how do we solve it? So first we have to calculate the concentration of the potassium in this 5 microliter. Okay, so the concentration that the instrument gave is 25 nanogram and it's for 5 microliter. So it will be 5 nanogram per microliter. Okay, so this would be the concentration that we have measured in this diluted sample. Now, this 5 nanogram per microliter, because we have already diluted the drug formulation 10 times. Okay, because 10 microliter of the drug solution and 90 microliter of the sample that we use, the solvent. Okay, so this dilution is 10 times. So we'll multiply with the dilution factor. So this would yield 50 nanogram per microliter. Okay. So any doubts till in this question? Okay. If not, then we'll move on to the next question. The concentration of pure water is. Can anybody guess? Okay. So we have got one answer, that is option number second. Then any more guesses? Okay. So yes, the correct answer is option number two. And how do we calculate it? We have the molecular weight of water as 18 gram per ml, or whatever unit you have to like. So 18 would be the molecular weight of water. So now in case of molarity, it's 18 divided by, and yeah, so it has a density of 1 gram per ml. Okay, so based on this, we'll calculate the molarity. So okay, so we have 1 gram of water in 1 ml. So based on that, if we calculate the number of moles, it would be 1 gram, and then 18 is the molecular weight. And here we have 1 liter solution, so this will be 1000 ml. So after calculating, it would be 55.5 molar. Okay, so this is how we calculate the molarity of pure water. So any doubts in this question? If not, then we'll move on to the next question. The concentration of hydrogen ions in pure water is, take a guess. Okay, option number 2. Anybody else? So yes, the correct answer is option number 2. And how do we calculate this? So first, we have to check the concentration of H2O is 55.5 molar, and the K equilibrium. Okay, that is basically the dissociation constant of H plus and OH minus. So K equilibrium is equal to H plus the concentration into OH minus upon H2O का concentration. So this is the value is 1.8 into 10 to the power minus 16 molar. Okay, so this is at room temperature, that is 25 degrees Celsius, where the pH of pure water is 7. This value is variable based upon the temperature. So here at pH 7, we'll at 25 degrees Celsius, this is the K equilibrium. So now if we have to check the product, this would be 1.8 into 10 to the power minus 16 into 55.5. This would yield 10 to the power minus 14. This product that we call as Kw is 10 to the power minus 14 molar. Okay, and out of these at pH 7, both of them will be in equal concentration. So this would be 10 to the power minus 7 and this would be 10 to the power minus 7. So this is how the concentration of hydrogen ions in pure water would be 10 to the power minus 7. Okay, so any doubts in this question? If not, then we'll move on to the next question. So we have to find out the pH of solution that contains 0.00001 molar OH minus concentration. So the OH minus that we have is 1 molar, or basically 10 to the power minus 5 molar. So what can be the correct answer for this question? Okay, any more answers? So the correct answer is option number 2. Okay, so there are two ways to solve this question. First way is the in the form of concentrations. Okay, so we have OH negative concentration as 10 to the power minus 5. Then, based on the formula that we just discussed in the last question, this is 10 to the power minus 14. And then putting these values, this would be 10 to the power minus 9. And if we have to find the pH of this particular solution, pH of this particular thing, that would be the negative logarithm of the hydrogen ion concentration. So this would be 10 to the power minus 9. This would yield us 9. Okay, and the second thing is we can find POH first. This would be 5. Okay, and then using the formula 14, sorry, so pOH is 5. That means pH would be 14 minus 5 is equal to 9. Okay, so any doubts in this question and how to apply the formula? If not, then we'll move on to the next question. So the enzymes works optimally at pH range of 1.5 to 1.6. So which of these enzymes that are given work optimally at a pH range of 1.5 to 1.6? Can anybody guess? Okay, someone said pepsin. That is option number 3. Any more guesses? Okay, so yes, the correct answer is option number 3, because as we all know, pepsin is present in stomach. Okay, and stomach usually functions at a pH range of 1.3 to 1.7. That is a really acidic pH. This helps in digestion basically. So pepsin works in this particular pH. Okay, so in case of stomach lipase, we do not have this like consideration. Okay, because stomach lipase is a different enzyme, and I guess there is one more reason that I'm not remembering right now. Okay, I'll let you know after some time. But pepsin is a major enzyme that have basically histidine and aspartate at their active site. Okay, so that requires an optimum pH of 1.5 to 1.6 for their catalytic activity. So this is the reason. I'll let you know about why stomach lipase is not the answer. Okay. So any more doubts related to the question? If not, then we'll move on to the next question. So buffering system that maintains a physiological pH of 7.35 to 7.4 in blood is triacetyl phosphate buffer, bicarbonate buffer, or acetate buffer. So someone said 3. Then any more options? D. Yes, the correct answer is option 3, that is bicarbonate buffer. So this basically is due to the carbon dioxide that we exhale. So in case of our blood, what happens, because our blood is mostly water. So when carbon dioxide is dissolved in blood, this forms H2CO3, that is a carbonic acid. Okay, and this being a weak acid, it's a buffer. So it do not dissociate fully and leads to plus H plus. Okay, so this equation leads to the buffering capacity of carbon dioxide in our blood. Okay, maintaining the pH of the blood between 7.35 to 7.4. So this is how carbonate buffer system basically acts on. Okay, let's move on to the next question. So in case of diabetes and fasting, dash is released which causes acidosis in stomach. Can anybody guess? Hydrochloric acid. Okay. Someone said HCl. Any other answers? So the correct answer is option number two, that is beta hydroxybutyric acid. So what happens is, in case of diabetes and fasting state, so what happens, the carbohydrates or glucose basically, for example, is like very limited. Okay, because in case of fasting state, more we are not taking any carbohydrates, so body is devoid of glucose. So now, as you might know, that glucose is the first preference for energy source. Okay, in the absence of glucose, the body shifts to the lipids, that is the second source, but it yields more energy than the carbohydrates also. Okay, so now when glucose is deficient inside the body, then lipids are the second source of energy. Then what happens, while metabolizing the lipids, various keto bodies are formed. Okay, so this is one of the ketone body only. So other ketone bodies are acetone and like the derivatives of acetone, like hydroxyacetone and like that. And the third one is the beta hydroxybutyric acid. So these ketone bodies are high in these patients where the lipid content is more, or the when lipid is being utilized as a energy source more as compared to carbohydrates. Okay, so this is why we see high amount of beta hydroxybutyric acid when we are fasting or when we are diabetic, because then also the body is not able to utilize the glucose that is present in the blood. Okay, so this is the correct answer. Any doubts in this question? If not, then we'll move on to the next question. Okay, so now we are moving on to the buffer system. So the conjugate base of an acid Ha, so this is a hypothetical acid Ha which dissociates in a buffer system as follows: Ha plus B negative gives A negative plus Hb. So what is the conjugate base in this case? Can anybody guess? Okay, someone said third option, that is A negative. Anybody else? So okay, anybody? Someone also said three, that is A negative. So what happens? We have certain acid-base systems. Okay, the concept first was the Arrhenius concept that basically tells that anything that gives H plus is an acid, and anything that will give OH minus is a base. Okay, then came the Bronsted-Lowry theory. So what does this suggest? That anything that will give H plus will be a acid. Okay, and anything that will take up H plus will be a base. Okay, so here if Ha would give H plus, then it would be an acid and it would form another part that is a conjugate base. Okay, so here A minus is a conjugate base of Ha, that is a acid, and this acid conjugate base pair forms a buffer. Okay, so what happens when acid is weak? It has a strong conjugate base that makes it a good buffer system. Okay, so here the Ha will release H plus and it will form A minus, like we have seen here. So A minus will be a conjugate base to Ha. So the correct option is option number 3. Okay, so any doubts in this question? If not, then we'll move on to the next question. So, during titration of a weak acid with a strong base, which of the following will be observed? So, the options are: pH will keep on increasing constantly, pH will increase rapidly initially and slowly near pKa values of an acid, pH will not increase, or pH will increase slowly initially and sharply near the pKa values of an acid. Can anybody guess? Okay, someone said 4. Any more answers? Okay. Any more guesses, answers? Okay. Someone said 2. So basically, what happens? We are making a curve with pH and the pK values. Okay. So what happens when we are titrating a weak acid with a strong base? So what we'll do, we'll take weak acid and then we'll add dropwise strong base to it. So this would increase the pH of the solution. Okay. Now, for example, the acid is at a pH of say 2. just hypothetical situation. Now you start adding the strong base. So what will happen? You keep measuring the pH after every addition. So the pH will sharply increase first. Okay. Then what will happen? There will be a decline in increase in the pH, and that would be slow increase in the pH. And there a point will come where it will be sharp increase in the pH again. Okay. So this region is called as the buffering region of a weak acid. Okay, and this region is near to the pKa values of that particular acid. So now, for example, if the pKa values of this particular weak acid is around 7.4, so here from 5 point something to 8 point something, this will be the buffering action. And here the pH increase will be very slow. Okay, because here the weak acid will start its buffering action and it will resist the change in pH. And initially the pH increase would be rapid. Okay, so this is the basic titration that we follow, and the correct option would be option number 2, because near the pK value the buffering action is active at that point of time. So it will resist the change in pH, hence the pH change would be very slow, and it would take much time to reach from this particular pH to this particular pH. Okay, so the correct answer is option number 2. So any doubts in this question? So if not, then we'll move on to the next question. So this is the similar type of questions that have been previously related to the previous question. So the titration of acetic acid with NaOH, the rate of change of pH in a range from pH 3.76 to 5.76 is slow. The decrease in rise of pH value in this specific range is because the options are: pH of solution approaches close to pKa values of the acetic acid, solution is saturated with NaOH, pH of the solution approaches close to the pK value of NaOH, or pH of solution approaches close to the pK value of H2. So, can anybody guess? The answer has been discussed in the previous question itself. So, yes, the correct answer is option number 1. Okay, and here is the explanation, the same similar explanation that I gave to the previous question. So while titrating acetic acid with the NaOH, the pH will start sharply increasing initially, then near the buffering region that is near to the pK value, the the pH will rise very slowly. Okay, so because it reaches the pK value of the acetic acid, then after covering this buffering region, the pH will again start to increase rapidly. Okay, so I hope the explanation is fine. So let's move on to the next question. So the dissociation of phosphoric acid, that is H3PO4, is shown below along with the pK values. So as it has three hydrogen ions, okay, so it will have three pK values because it can dissociate all the three ions. So which of the following pK values makes it a suitable phosphate buffer system in the pH range of 2.9 to 3.5? So the options are PK1 and PK2 both, PK1 alone, PK2 alone, or PK1 and PK3. I guess this is the wrong option, but yes, please guess. Okay, someone said option 2. Any more answers? Okay, option 3 also someone said. Any more answers? Okay, so yes, the correct option is option number 2, as I have explained in the previous question also. So the buffering region will be near to the pKa values. So here the buffering region is 2.9 to 3.1, that is near to the pKa1 values. Okay, so the correct answer would be option number 2. If the pH range would be near to this between 6 to 8, then pK2 would be the correct answer. Okay, and similarly with the pK3 values, if it's near to the alkaline pH, that is between 11 to 13, then the pK3 would be involved in the buffering action. Okay, so let's move on to the next question. From the Henderson-Hasselbalch equation, pK can be defined as the pH at which the options are acid is 100% ionized, acid is 50% ionized, acid is 99% ionized, or acid is 10% ionized. Can anybody guess? Okay. Yes, the correct answer is 50% ionized. So, actually, what is the Henderson-Hasselbalch equation? So, it's basically pH is equal to pKa plus log of the electron acceptor upon the hydrogen donor. Okay, so when this concentration will be equal to this concentration, then that would be equal to log 1. Okay, and log 1 is 0. So at pH is equal to pKa, the both the acceptor and the HA or the donor will be equal in concentration. So that means the acid is 50% ionized. Okay, so this is how we get the pK value is at sorry pK value is at pH where the acid is 50% ionized. Okay, so let's move on to the next question. The pK value of an acid is 7.76 at 25 degrees Celsius. Therefore, the pH range in which buffer will exhibit high buffering capacity would be, the correct answer would be, okay, someone said option number 3. Yes. So the answer is option number 3 only. So the same explanation is the that I've been giving from the last questions. So the pH range in which the pK value is near. So 3.76 to 5.76 is not near to 7.76. So the option 1 is wrong, similar to the option 2. But for option 3, 6.76 to 8.76, here the pK value lie in between. So the buffering action will be active here. Okay, so the correct answer is option number 3. Okay, then we'll move on to the next question. From the given chemical reaction, that is HNO3 plus H2O gives H3O plus NO3 minus, the Henderson-Hasselbalch equation would be, how it can be written? Can anybody guess the answer? Okay, someone said option number 1. Any more answers? So the Henderson-Hasselbalch equation suggests that pH is equal to pK plus log of, so hydrogen acceptor upon hydrogen donor. So in this equation that we see, the pH will be equal to the pK plus log of the hydrogen acceptor here is NO3 minus, and the donor would be HNO3. Okay, so the option 1 is correct. Okay, then let's move on to the next question. Which of the following acid is recommended to adjust the pH of a freshly prepared phosphate buffer to 7.4? So, can anybody guess? We are preparing a phosphate buffer. So, which acid will be using to adjust the pH? Someone said option 1, that is 1 normal H3PO4. Okay, any more answers? Okay, so the correct answer is option number one. Why? Because the H3PO4 that we see will yield us phosphate ions. Okay, and we have seen in the previous questions also, at all the 3 pK values it can show the buffering capacity. Okay, so that can be involved in the buffering capacity of the phosphate buffer as well. So the correct answer is 1 normal H3PO4. Okay, so any doubts in this question? If not, then we'll move on to the next question. What will be the pH of the acetate buffer if concentration of acetic acid is zero point 0.1 molar and sodium acetate is zero point 0.2 molar and the pK of acetic acid is 4.76? So this is directly based upon the Henderson-Hasselbalch equation. So can anybody guess? Okay. Any more answers? Someone said option 3. So the correct answer is option number 3. So how we'll solve it? The Henderson-Hasselbalch equation for acetic acid acetate buffer would be log acetate ions and the acetic acid, because this is the donor, this is the acceptor. Okay, so we have to calculate the pH. pK is given 4.76. For acetate ions, we'll see sodium acetate because this is a salt. So while dissolving, it will directly dissociate to sodium ions and the acetate ions. So we'll directly put the concentration like this. And for acetic acid, we have zero point 0.1 molar. So after calculating, we'll get 4.76 plus log 2. So log 2 has a value of 0.301. Okay, so this would yield near to 0.05, 5.06. So the correct answer is option number 3. Okay, so just a general suggestion to everybody, remember these 4 log values for your reference for any competitive exams, for general calculations. log 3, log 2, that is 0.301, log 3, that is 0.477, log 5, that is 0.699, and log 7, that is 0.845. So remember these four log values. This will be very helpful in your any calculations related to log, or in any of the exams that you give. Okay. So any doubts in this particular question? So if not, then we'll move on to the next question. So what is the pH of the solution resulting from a mixture of 200 ml of zero point 0.1 molar NaOH and 100 ml of zero point 0.3 molar uh, acetic acid? Okay. Any more answers? Just a second, guys. Problem there. Just 2 minutes. Sorry, a problem again. Encountered. So yes. So the correct answer would be option one. So how will we solve it? First, we have to calculate the amount of salt that would be present. Okay. From that salt, what we'll do is we'll put in the Anderson-Hasselbalch equation, and then we'll calculate the pH of that particular solution. Okay. So here the correct answer would be 5.07. So the equation that it will follow will be plus. Okay, so from this equation we have to calculate the amount of or the concentration of sodium acetate, and from this we'll put it in the this particular equation. Okay, so please try to solve it. Okay, and then if you encounter any problem in this, we'll discuss in the next class. Okay, because due to the time constraint, I'll not be able to explain this particular properly, but we'll discuss in the next class that how will it will be solved if you guys encounter any problem. So move on to the next question. So this was the last question basically. So any doubts in this particular presentation or any of the concepts that have been studied in week one, we can discuss it now. Any question you are not able to understand or any concept we can discuss. if no doubts, uh, we'll close the session for today, and we'll meet on next Saturday with the discussion on week 2. Okay, so thank you guys for joining. Thank you.